truly exciting to see how a disease that was a death sentence for the patients is now getting its therapy for it. The next speaker is Miguel. Please.
Thank you very much. Okay, now it's my turn. Thank you, Batsheva. And Batsheva is here, and we need to thank her very much because she just became another time a grandmother. So thank you very much for being here and for coming. And I want to probably shorten my talk since we are all hungry. And I want your attention as well. And I would like to show you, probably we will skip on a lot of slides, but I would like to pass on a new approach, and we call it a road towards drug-personalized medicine of rare diseases, and how it works. So this approach is based on image high-content analysis of cells. These cells are coming from obviously from patients with rare diseases, and that's kind of a basis for our personalized medicine approach. And we would like to prepare and find a way to do analysis on patient cells, identify their specific phenotype related with the disease, and by doing so, do a personalized screening drug discovery for those patients with the rare diseases. So our platform would like to answer to several of those questions that were addressed before me, which is based on how we do it, how we overcome diversity, which also exists in the rare disease population. Even those patients that have the same mutation for a certain disease have different outcomes. And on the other hand, probably the personalized drug discovery approach could hamper that kind of problem. But obviously, we're just on the beginning. And to do so, we need to identify what is a platform, on which type of cells we can do our work. And this is based on, and I don't know why this is not working. Do we have Avi? But it doesn't move my way. Okay, it doesn't matter. The most important thing that is we are working on the models of disease. So which type of cells are those that can be used for screening purposes? And those are that obviously you can take one sample out of the patient and then you grow the cells by thousands in the lab. So therefore, you are postponing kind of a platform that you have these cells with a proliferate capacity that on the other hand, these cells still need to be keeping their phenotype, their basic phenotype, the basic cell-based cell type that will not change with time. This capability of proliferation will allow you two things. One is drug discovery, drug screening, which allows that you consistently could do your screening procedures not only once, but many times in order to validate your result under the same exact experimental condition, which is an important asset. And secondly, this could be due for personalized medicine because obviously you're coming from cells that are directly isolated from patient samples. Okay, having said that, and we thank you, Neta, for talking about personalized medicine. So I can skip now this slide. Or what is the meaning of personalized medicine? Is basically getting the right pill for the right person and get to the right drug, right dose on the right time. So basically, the problem is that although it sounds fantastic, Neta also put it together that it's kind of looking for a needle in a haystack. So how do we go through this problem? So obviously, a lot has been said today about individuals' genetics, about the technology based on the mutations, and probably in the future we will know much more about all these mutations and the relevance to the disease that what Duron was talking about today. But our technology is based, and I will go through this fast. These pyrotechnics we don't have the time for that. So basically, we are trying to find a bottoms-up approach. Identify the phenotype. The phenotype, the cell-based phenotype, is telling us, the cells are telling us that their biology is on. So basically, by analyzing phenotypic information coming from cell-based analysis of images from cells coming from patients, we will probably be able to identify the phenotypes that are related with the disease, and these phenotypes are multiparametric, which are basically identified by probably a play of thousands of genes coming together. So by looking bottoms up, we are probably telling how things go wrong, and probably the cells are telling us that their biology is different. How we do that? We culture by thousands in multitator plates, 96, 384, for example, thousands of cells. This can be easily imaged by image analysis using this high-content microscopy analyzer, which photographs labeled cells, so we label them fluorescently. We have 4 different channels of fluorescence intensity, so we can label different and simultaneously many targets. In a way, you could analyze each one of these thousands of cells that are being scored in thousands of images, and then you do it automatically, and then you could come up with important numbers out of those images. Once you have numbers, you can measure, and you have on your desk thousands of numbers that are telling you, this is our biology, this is the biology of the cell. Now our task is to differentiate and to identify these thousands of data coming up from these images to tell how rigorous and how robust these differences are, enabled to understand that we have an assay for high-throughput screening, which is, for example, a disease-based phenotype without knowing the etiology. So we could be able to screen cells with a disease, even without knowing the etiology of the disease, which is most relevant for rare diseases. And obviously, how this works. So, and this is the power of the analysis, of the methodology. So here we have the most complex cells to analyze in image-based analysis, which are neurons. Okay, so this is only one row of thousands of rows, thousands of images in one Excel file. So here we have a couple of hundreds of neurons, and the machine can identify each cell under 4 channels and who is talking to who and so forth. So our idea is, okay, all this information is gathering up. We need to understand what is the meaning of this. This is for you to see how sensitive the machine or this methodology can be. Okay? So we started about 6 years ago using this technology. We are the first ones in Israel to get this type of technology of high-content analysis. We managed to incorporate our automatic robot. We did some screening for adult polyglucosan body disease with 20,000 compounds. Or Kahlon will talk more about the details of this screening, and we come up with nice hits for this, coming up from cells coming from patients, using this methodology. Now, we were lucky that we were working on these several diseases using exactly the same approach. So the idea is we are building a model that not only will work for one disease, that probably this will work, but for many, or for at least in parallel for each one of these rare diseases. And now having the opportunity to to bring in this technology to the Blavatnik Center for Drug Discovery, we will now are in a position that not only we will be able to screen as we did it before, now we will be able to adapt and probably move on in where most of the screening processes can start, and people that are there are many labs that have been screened before and have got a hit, and therefore it will take a lot of time for them to move on to the next step. In our case, and for others in the audience that are in the same position, could come here to the Blavatnik Center and try to move on to the next step of drug discovery. Okay, and this is for me probably the most important take-home message for you, and to know what we can do today regarding using this technology. Okay, and the ideas comes like this. So I mentioned that we work with patient cells coming from patients with different types of rare diseases. The idea, once we have culture and be able to make them grow in culture, do the analysis, the image analysis, get a profile, basic profile based on image analysis, and then try to understand what the meaning, the biological meaning of this difference might be. Because you could see that the parameters, as we show you in a second, how these differences look like, but we don't have a clue what the biological meaning is all about. Okay, what is behind? Which pathways are behind this morphological, let's say, image-based differences? So we want to do, in parallel to this, after photographing them, we could take the profiles, extract using high-throughput manners, mRNA, microRNA, and even do metabolic profile of each one of the cells, of thousands of cells in parallel after doing the imaging. The idea is after doing the imaging, we could be able to integrate this information, and that one of our products that we would like to produce is do and help the rare disease community by adding value that probably will have some clinical value, which is those informations are coming up from the cells themselves, how they behave, and probably get a better perspective of their diagnostic and prognostic abilities of these cells, enabled to classify in the future those patients that probably are outliers also in the community, specific community of this rare disease group, for example, in a way that probably we will be able to classify the phenotypes of these cells, and could in the future could predict that this lookalike cell type is part of this type of this kind of patients with this type of clinical outcome and so forth. So this is one of the power of the disease of our platform, and the other is more long-term and most difficult to make, which is based on using our capabilities of phenotyping using drugs. The drugs might tell us which pathways are being specifically changed or specifically modulated or perturbed by the drug itself. So without any based hypothesis, we could able, as I spoke before, screen the cells without knowing, and by analyzing the profile, which is now integrated with mRNA, microRNA, and metabolic profiles all together, and see how the effects of such compound might have an effect on the phenotype of how the cells look like, and we might be able to understand how molecularly, or which might be the active targets involved in these changes. So that's our idea, and at the end of the process, we would like to come up with a drug candidate that will have a more future importance for treatment of the specific disease. So that's what we can do. I don't have time to speak about why and how long we've been doing with this system of phenotyping and biomarkers identification, but this starts 12 years ago using ALS cells coming from ALS patients, which are non-neuronal. These were bone marrow mesenchymal stem cells, and we managed quite nicely to identify 4 specific or ALS-related biomarkers, which were found to be also abruptly expressed in relevant tissues of the nervous system and muscle of the mouse model, for one of the ALS mouse models, which strengthens the idea to come out of the box thinking, which is we started with cells coming from patients with ALS, which is a neurodegenerative disease, but these cells are non-neuronal. So this gives us the power that we were looking probably on a nice model of the disease, but now coming from humans. And well, to make a long story short, this is a work of Shane Weil that she just got her PhD today. Thank you very much. Mazal tov. She was confirmed today. And here we have an idea. We treated mesenchymal stem cells come from 5 different ALS patients. All have different type of ALS, obviously, because ALS is a multifactorial disease. And as you can see, we induce them with a double-strand break, makes the DNA break up. And then, usually after this treatment, a normal cell will repair the DNA, and the impact on the viability here in blue, you see that this induction does not affect the normal population, while in the ALS population we have a shift. What does it mean? Well, it means that the ALS cells have something different in the response to DNA damage regulation. Okay, to make a long story short, that comes regarding with markers known for autophagy. This is a kind of a response of the cell which is specific for the ALS, and it doesn't happen in the healthy control population, which induces some kind of markers that we know that are related with autophagy. It comes up at 24 hours, and this is not working anymore. And come up also together with the lysosomes, which are associated with the effect of autophagy that comes up after 24 hours of this treatment, also in this ALS population. But most importantly, we can block this using early stage autophagy inhibitor called 3-methyladenine, which can block this cell death inhibition. I can tell you that blocks also the autophagy, and the cells are fine. So the idea here, the model, it coming from bone marrow mesenchymal stem cell. It is making us a point that they are showing you can put them under certain stressful condition. You can find a mechanism of disease based on which is specific or not specific, but different in the ALS population compared to controls. So this makes a good model, and now I would like to stress the power of understanding. Okay, so now we have cells that are from ALS patients that show that they have biological meaning, differential activities, and now we want to see how this translates into the phenotypic analysis that I've been talking about. So we can even label simultaneously the lysosome, the cytoplasm, the mitochondria, the nucleus of the cells, and do this parameter analysis. And these are differences between ALS in red and green. We have the healthy control population. These are thousands and thousands of cells have been screened, and in an average of 4 ALS patients and 3 non-ALS patients. What you see here is the histogram of how the cells behave in the mitochondria. This is only one parameter. We can produce hundreds of parameters, and or will explain to you later on more about these parameters. But here you can see in green that the median the average median of that you can see a differences between how this population shifted in between ALS and no-ALS, also in mitochondrial and lysosomal. Like this, we have dozens of different parameters that are showing that the ALS population, cell population, is different in some way compared to just by analyzing images. Okay, so how does it work? So we have here a kind of measurement, which means that we could analyze statistically all these thousands of cells coming out from this information, and now we have bars that you see in the red, which is the Z-score, which is the difference of the standard deviation of the population, of the ALS population compared to healthy. So what we see here is how different the ALS is from healthy. Okay, here we have kind of a profile of how the cells look like translated in numbers. Okay, what you see is how this is significantly different from healthy controls. At the nuclear cell, at the cytoplasm, different features that are coming out from there, mitochondrial and lysosomal. These are about 30 different, 25 or 26 different features. What you can see here, and this is the nice thing about how personal you can get here in this analysis, you can identify in the red spectrum, you could see the ALS, and the blue is the healthy. What you could see, the healthy are closer to zero, while the ALS tended to be far apart. And these are each individual. It's analysis of cells, profiles from each individual. Okay, so something is there. We still don't understand what does it mean, and whether this could be a useful way for screening drugs for ALS. This is early days. But this means that we could, just by analyzing images of cells coming from patients, we could identify potentially a patient-specific, the disease-specific profile. Okay, and to make a long story short, we also did it for other diseases. We need to throw a... Okay, and what you can see here is just raw data showing an histograms of 35 different, I cannot see from here, 20 different features coming up from mitochondria, nucleus cell, and lysosomes. What you see here is a heat map of the diversity of how this is spread, like an histogram of showing simultaneously different histogram of 20 different features of showing 3 patients with DMDs, Duchenne. Each is a patient with a different mutation. APBD, which is the glycogen storage disease that Takehome will talk about later on. And MNG, which is a metabolic disease, all rare diseases, compared to wild type. Wild type, we call wild type. We call wild type, obviously humans don't have a wild type, but this means healthy control. And you can see that, for example, between the Duchenne, there is a similarity in the profiles compared to the other 2 diseases, and obviously very different from the healthy. Now, another way to look at, for example, the lysosome. The lysosomal parameters show some similarity between all these diseases, which is probably significantly different from the healthy. So looking at this, the idea is we have multi-parameters coming up from thousands of cells, and now this is one way to see how things where we have the difference and where we have the similarities, and how a patient could look like similar to other with the same disease or different from other diseases. So this is a basic way to analyze your data. We still don't know what the significance of this data is, but yet this is a kind of an easy way, because we are talking about raw data. We haven't done anything to do a further analysis. It's just how the cells look like. So this is an interpretation of the images as easy as it gets. Okay? So, how much we have? -2. -2, okay. So to make a long story short, and probably here in the audience, you know about familial dysautonomia, which is caused by a mutation in the ICAP-BCAP gene, and it's an also autosomal recessive disease from Ashkenazi Jewish population. And we've been, there is a small molecule called kinetin that have shown to have beneficial, at least in vitro and in some animals with model of disease, and has been treated also, as some clinical trials have been done with kinetin, and show that this drug can fix the alternative splicing that is occurring here. In here we have neurons coming from, we developed a system using human embryonic stem cells with FD, with this mutation that come from a prenatal genetic diagnosis clone that was originated in Ben Rubino's laboratory. And we treat these neurons now from embryonic stem cells, we transfer to neurons, and you can see that this kinetin can fix the alternative splicing. The meaning for the neuron, it takes around 4 to 8 days. Sorry. It takes around 4 to 8 days to bring back the protein in place, and you can see in green the ICAP before and after treatment is back in the neuron. Okay, together with ICAP, there are coming other important proteins that are relevant for neuronal function. I don't have time to get into this, but the most important thing is this drug works on neurons and in alternative splicing. Now we wanted to see whether our system, using now fibroblasts of FD patients, and now we are showing only one example. Here we have how the alternative splicing is occurring in the FD. We have a carrier, we have a healthy control, a control without the mutation. You could see after 8 weeks, we are now trying to find how the phenotype is influenced by the kinetin, the cell phenotype, not only the splicing, with whole cell phenotype. So here you see it restores. If you remove the kinetin 1 week before the 8 weeks ends, it comes back. Now, how the profile looks like. In red is we have here below, we have all the parameters I'm talking about from different places in the cell. In red, we have the FD. In blue is the carrier, and green is the wild type. You can see that the FD shows lower signature compared to the others. If we put back if we have treated with kinetin, it looks, everything goes up. So that is for us a kind of a proof of principle that this system of wide multiparametric analysis can work, that we use one agent that is known where it works and how this gene has an impact in the overall phenotype population. This is just a proof of concept. And to make a long story short, I want to thank all my people in the lab, the funding agencies, my collaborators, and to you for your attention, and obviously to Nir, who is sitting here, for giving my everything. And now lunch is served. Thank you.